Today we're going to be talking about mitochondrial mutations. Now, in order to understand these, first we need to talk a little bit about DNA, what it looks like, what it's made of, and how it fits together. Nucleotide bases fall into two categories based on their chemical shape. The first are purines. These are double-ringed structures, and for nucleotides, these are adenine, guanine. The second are pyrimidines. These are single-ringed structures, composing thymine and cytosine. These bases are connected to the phosphate backbone in pairs, with one half of each pair on each strand. So, for example, you might have adenine on one strand connected to thymine on another strand. Because they occur in bases, we call them base pairs. Because of their chemical structure, these base pairs connect in a unique way. Adenine always connects to thymine, and guanine always connects to cytosine. If we take this helix structure of DNA, we stretch it out so that it's in a straight line, it's going to look something like this. We can see that there's the two strands on the top and the bottom. We call these the forward strand and the reverse strand. It's not that one's facing forward and the other one's facing backwards. It's just a way to differentiate the two. So we can see here at this first location that we have the nucleotide A on the forward strand, the nucleotide T on the reverse strand. Together, these form the base pair AT. We can see right next to that that we also have a base pair AT, but this one's a little bit different because instead of the A connected on the forward strand, we have the T connected on the forward strand. This is going to become important a little bit later on. To make things a little bit easier for both testing and reporting, we separate out the forward strand and the reverse strand, and we only report the nucleotides that we find on the forward strand. So, for example, on the first location, we would simply report A, not AT, because we know that A is always connected to T. So by reporting A, we automatically know what the nucleotide on the reverse strand will be. In addition, we assign each location a number so that we can better identify where they are in the genome. Now, there are millions of locations throughout the genome. So if we're trying to find a specific sequence or a specific area, it can be a bit like trying to find a needle in a haystack. So say, for example, we only want to look at locations 3 through 22. Well, how do we pinpoint that? Well, the way that we do that is we essentially create artificial sections of the reverse strand that we call primers. So in this example, we create an artificial primer that has a TA. And this we know is going to pair at locations 1 and 2. It's not going to pair anywhere else because, again, T only connects to A. So we know that even though we've separated these two, that this primer isn't connected to the reverse strand. It's only going to connect to the forward strand. In reality, this primer is going to be a lot larger so that it doesn't get mixed up with other areas of the genome that might just randomly happen to have an AT. But for demonstration purposes, let's just say that it's simply the artificial T and A. And then we also create the artificial T and G because, again, we know that it's going to lock in here at locations 23 and 24 on the forward strand. Now, we do something special with this as well. We add a fluorescent dye so that when we run it through the machine, it kind of highlights. It says, hey, look at me, I'm the primer. Here's the start and end points. You want to look in between everything here. This is the area that you're going for. So that way, when we see it, we can say, okay, now everything that we test in between these two sets of primers is the area that we need. We can also verify the results of this test by creating primers specifically for the reverse strand. So we do the same thing, create primers between locations 3 and 22, but in reverse. We just make an artificial AT and AC. We add a different kind of fluorescent dye so that it tells the system, hey, I'm a reverse primer, not a forward primer. And then when we read the results in between that, the system will automatically say, oh, okay, there's a T here on the reverse primer. That means really we need to report this as A because it's a TA pair. Now that we understand a little bit about the structure of DNA, let's talk about mitochondria. Now, mitochondria are special organelles that exist outside the nucleus in a cell, and they actually came along very early on in evolution. And it's theorized that they were originally a type of bacteria that formed a symbiotic relationship with cells. Basically, they came into cells, and these cells provided a safe, secure environment inside their membrane for the mitochondria to flourish. And in return, the mitochondria allowed the cells to utilize the oxygen all around them as an extra source of fuel to reproduce, replicate, and form all the life forms that we see today. So if it wasn't for mitochondria, we wouldn't have any sort of multicellular organism. Now, when mitochondria first came into cells, it brought its own DNA with it. And this is still DNA that's in the double helix structure composed of the 4 nucleotides, but it's in a little bit different of a structure because it forms a ring. The ring structure of mitochondrial DNA is composed of 3 main regions: HVR1, HVR2, and the coding region. HVR1 and 2 together form only a small portion of the overall mtDNA genome, and the mtDNA genome in turn is considerably smaller than the DNA that we find inside the nucleus. When you unhook that ring and you unravel the helix structure, you get the same basic shape of these 2 strands that are connected by base pairs. And again, each one of the base pairs has a location. This location system is separate than the one used in nuclear DNA because it's its own genome. And again, for testing purposes, we only need to focus on the forward strand, so we're going to go ahead and ignore the reverse strand. Now, this sequence, when it was first created by a group of scientists at Cambridge University, each one of these locations was assigned a value, which we call an ancestral value. So it was standardized, for example, that at location 1, there was the nucleotide A. At location 2, there was the nucleotide T, etc. If a mutation occurred and we saw something that differed from our reference sequence, such as an A turning into a C, then we called this a derived value. The very first mitochondrial reference sequence was developed at Cambridge University in the late 1970s and early 1980s and was known simply as the Cambridge reference sequence. Shortly after it was released, they noticed that there were some values on a few markers that weren't really representative of the population as a whole, so they made a few slight adjustments and they released what is known as the Revised Cambridge Reference Sequence, or RCRS. And the RCRS has pretty much become an industry-wide standard for measuring mutations. This was based on a group of anonymous individuals of European descent who were affiliated with Cambridge University. And in the RCRS, the ancestral values are also known as reference values, whereas the derived values are also known as mutations. And the way that we report these mutations are the location of the nucleotide plus its derived value. So, for example, if the ancestral value at location 263 in the chromosome is A, then we report it as 263G if the mutation has G. Now, as I mentioned, mitochondria is broken down into 3 different regions. So we report these mutations by region. So on this first one here, you can see that at location 16192, there was a mutation or a derived value to T. Now, you might notice something slightly different here at this marker, where it's followed by a Y. Now, obviously, Y doesn't fall into the category of the A, C, T, or G that we find in DNA, and this instead represents a special kind of mutation called a heteroplasmy. To understand heteroplasmy, let's take another look at the cell. Now remember that mitochondria is outside the nucleus. There are actually many mitochondria in each cell. And these don't always have the same markers in the same locations. They don't always mutate all at once. Sometimes you'll find that some mitochondria have mutated in a particular location, while others haven't. This is essentially a mutation in progress. It's sometimes gradual and sometimes might take a few generations to fully change from one value to another. So for example, in one location, if we have a value of C in some of the mitochondria, but at that same location we have a value of T in some of the other mitochondria, then this represents essentially a mutation in progress. So we don't know if it's T changing into C or if it's C changing into T. Because sometimes, even if a mutation has happened from the reference sequence in a previous generation, it can mutate back to that reference sequence in a later generation. So there's really no way to tell. So instead of reporting it as C or T, we report it with a special character that represents that it could be one or the other. So in this example, because we saw Y represented on that one marker, that Y means that it actually is either C or T. And the chart on the right here explains what the different symbols that you might see are. And you can find this chart and other information in our Help Center. Now we know that when we see a location followed by a letter other than A, C, T, or G, it represents a heteroplasmy. In this case, it represents either a C or a T. Now there's a couple other things you might notice here. One is that this location has a decimal point in it, and this one has a dash. These are two types of mutations known as insertions and deletions, commonly abbreviated as indels. First, let's talk about insertions. Insertions occur essentially when an extra base pair wedges itself in where it doesn't really belong. So, for example, let's say that right next to the base pair of AT, we get another AT. Now, this one doesn't really belong there, so it doesn't really get its own location. Instead, we just sort of represent it as the same location number, but followed by a decimal point. So instead of 315A, it's 315.1A. Next, let's talk about deletions. Deletions are basically the opposite of insertions. It's when a base pair should be there, but is missing. It's just, instead of wedging itself in, it's just been wedged out. Now this one doesn't really have a derived value, it just has no value. So instead of followed by a letter, we follow it with just a dash. So instead of 522, it's just 522 dash. Now that we understand the different types of mutations, let's take a look at the mutations as a whole. Since these are all differences of the reference sequence, what we're able to do is compare the specific mutations that we see in a group of people and compare it to another group of people. So for example, we're able to compare the mutations that we see in a European population as opposed to an Asian population, track those differences, and then be able to see to some degree how those two groups split apart. Because when they split apart, they took their mutations with them and they started developing their own unique mutations. And when we look at all of these different mutations together, we're able to trace together migratory routes called haplogroups. The oldest humans that we know of lived in Eastern Africa hundreds of thousands of years ago, and from there they spread all across the globe. And as they split apart, they started to develop their own unique set of mutations that were different from other groups. So each one of these migratory groups we call a haplogroup. And these different haplogroups represent different sets of mutations that we identify with a letter, such as U, or H, or M. Now you may be wondering why a group of modern-day Europeans was used as a reference population if the oldest humans lived in Africa. And that's a very good point, because as time went on, we started to see that some of the values in the RCRS weren't actually representative of the values that we see in those locations in the global population. So instead, a group of scientists created the reconstructed sapiens reference sequence. This was in a paper published by Dr. Daron Behar and a group of other scientists in 2012, and it was designed to represent mitochondrial Eve. In other words, a reconstruction of what the mitochondria in the earliest humans probably looked like. In the RSRS, we still have ancestral values referred to as reference values and derived values referred to as mutations, but they're reported a little bit differently. They're reported as the ancestral value, then the location, and then the derived value. So, for example, if the ancestral value was A at location 769, and we found a mutation to G, then we report it as A769G. And it still gets reported, broken down by the area in mitochondria where it's found. And we still have things like indels and heteroplasmy, but you may notice one slight addition, which is that some of these locations are reported with a lowercase letter instead of an uppercase letter. And this represents a special type of mutation called a transition. Transitions are one of two types of mutations that we find, along with transversions. And of the two, transversions are much more common. So first, let's take a look at these. This occurs when a purine is exchanged for another purine, or a pyrimidine is exchanged for another pyrimidine on the same strand. So for example, you might have an A on the forward strand replaced by a G, or you might have a C replaced by a T. Now, this is much more common because of the shape of the DNA. There's already a double-ringed structure in the DNA, so if a mutation is going to happen, it's more likely going to get replaced by another double-ringed structure because the shape is already there. And the same is true for pyrimidines. In contrast to transversions, we have transitions. And these occur when a double-ringed purine is replaced by a single-ringed pyrimidine, such as A being replaced by C, or vice versa. This is much less common than transversions because of their chemical shape. All of these mutations combine to form your haplogroup. Now, if you've ever seen haplogroup results, you'll probably find that it's more than just a single letter, such as H. It's probably followed by a series of letters and numbers. And these letters and numbers represent subgroups within the overall H haplogroup. Each one of these subgroups is defined by a mutation that's found in addition to the mutations found in overall H. So, for example, in this one, it will have all of the mutations found in H, as well as additional mutations that define its subgroup. There is one other type of mutation that I'd like to talk about, and that is a back mutation. Back mutations occur when a value mutates from the derived value back into the ancestral value. And we're able to determine this happens because we're able to see all of the mutations that define this haplogroup, but one of them just seems to be missing. Instead of the mutation we normally find, it has the ancestral value found in the reference sequence. So this means that somewhere along the line, a group of people in this subgroup had a marker that mutated back to the original value. And this is represented by an exclamation point at the end of the haplogroup. Whenever we find mutations that should be in a haplogroup but aren't, these are known as missing mutations. On the other hand, sometimes we find a mutation that isn't normally seen in a person's haplogroup, but is there anyway, and we call these extra mutations. These get reported in RSRS as extra mutations and missing mutations.